Excellent. Here's your intro to King 225. He wants to be the next Miles Davis, but without the heroin addiction. Okay, here we go. So, just a couple of announcements before today's class. Don't forget that your lab number 7 is due on Monday, and that link that I posted last day to children's physical activities, that story about Red Deer, Alberta, that was an April Fool's joke. So you don't have to know anything from that link that I posted last day on children's physical activity. So for today's class, I'm going to review fat metabolism, the glucose-alanine cycle, transcription and translation. I'm going to talk about the ways that satellite cells are activated and deactivated. Recall that satellite cells are important for protein synthesis and your response to resistance training. I'm going to talk about adaptations to resistance training, specifically with hypertrophy versus hyperplasia. So hypertrophy refers to, if you're talking about resistance training, it refers to increases in muscle fiber area or muscle fiber size. And hyperplasia refers to the increase in muscle fiber number. So which occurs with resistance training is a topic of debate. So I'm going to be going over that today. I'm going to also cover adaptations to aerobic training, and then how resistance training and aerobic training might interfere with each other. I'm going to go over the physiology and some training strategies to prevent that interference. So some review questions. Which metabolite can be used to make a triglyceride? The best answer to this question is all of the above. So a triglyceride is made up of 3 fatty acids and glycerol, but glucose can be taken up by adipose tissue, and it can be made into glycerol, and it can also be made into fatty acids. So the best answer here is all of the above. Which hormone is not involved in mobilization of fats from adipose tissue? Your best answer here is insulin. So insulin tends to pack away fats in adipose tissue, so it'll pack away triglycerides, whereas epinephrine, norepinephrine, and growth hormone, they're all released during exercise. They're involved in activating an enzyme called hormone-sensitive lipase that breaks down triglyceride to glycerol and your fatty acids. This is a review question with regards to fat metabolism, so a topic that we covered last day, and this is fat metabolism during exercise. And the review exercise is to put these steps in proper order. And so I've printed this off and review this with you. Just trying to put these in proper order here. So here we go. So the first step that you should identify here is release of those hormones that are involved in activating breakdown of triglyceride from adipose tissue. So it would be this would be your first step here. It would be release of growth hormone, norepinephrine, epinephrine. So those get released when you start exercising, and they'll act on the hormone-sensitive lipase at adipose tissue to break down your triglycerides. So your number 2 step here would be mobilization, the breakdown of triglycerides in adipose tissue into glycerol and fatty acids. So that would be your second step. Let's see. So that's over here, second step. The third step would be, let's see here. No, sorry, I got that messed up. First step is release of these lipolytic hormones, growth hormone, norepinephrine, epinephrine. Second step is activation of this hormone-sensitive lipase. And that is the enzyme that will help to break down triglycerides. So then you get this as your number 3 step. Okay. After that, your glycerol is transported to the liver and it can undergo gluconeogenesis. So I don't have that in these steps. But the fatty acids here, so the glycerol goes to the liver for gluconeogenesis, gets made into glucose. The fatty acids then have to go to the muscle, but they first have to be transported in the blood. So you get circulation, transport of fatty acids to muscle from adipose tissue. So your circulation is important here. If you have an increased capillarization, as you do with endurance training, then you'll have enhanced circulation of the fatty acids to muscle. So that's your 4th step here. The 5th step is the take-up of fatty acids into the muscle. So you get uptake of fatty acids into muscle. And I'll mention that there's proteins that are involved in the transport of fatty acids across the muscle fiber membrane. And those proteins get increased with endurance training. So you become better at using fats if you're endurance trained. Then the next step you get is you have to get those fatty acids into the mitochondria to break them down. So the next step is translocation, entry of fatty acids into mitochondria, and that's done by this carnitine carrier that we covered previously. So that would be your 6th step. The next step is the first metabolic pathway that the fatty acids go through in the mitochondria, and this would be beta oxidation. So beta oxidation involves the breakdown of the fatty acid by 2 carbons at a time, and each time you go through a round of beta oxidation, you get formation of 1 NADH. So that would be your next step. That NADH can then go to the electron transport chain, and it can help to make ATP. So specifically, it makes 3 ATPs in the electron transport chain. After beta oxidation, you chop up the fatty acid 2 carbons at a time. Each of those 2 carbon units will make acetyl-CoA, and those can then go through the Krebs cycle. So the Krebs cycle would be your 9th step in the process. And with the Krebs cycle, you form you form NADH. So then you can have this as the 10th step as well. And then the NADH can go to the electron transport chain to make ATP. So this would be your 11th step. I kind of jumped over this a bit, but when you get formation of NADH in beta oxidation, that NADH will also go to the electron transport chain. So you could have had, I'll just put it as 8B here as a possible sub-step in there with beta oxidation. So those are the steps from release of those hormones involved in activating triglycerides and adipose tissue, all the way to the oxidation of fatty acids in your mitochondria. Okay. Which of the following is not involved in transcription? The correct answer here would be your ribosomes. So in the nucleus, you have DNA, which encodes for your proteins. When you want to express a new protein, mRNA will transcribe a portion of the DNA. So it'll take a message for a protein from the DNA, and it'll get transcribed onto messenger RNA, which then takes that message outside the nucleus to make a new protein. So transcription involves DNA and mRNA. Which of the following is not involved in translation? The correct answer here would be DNA. So your ribosomes and mRNA are involved in translation. So when mRNA leaves the nucleus, it goes to a ribosome, and then it contains the message to make a new protein. Individual amino acids will come in and make that new protein depending on the message that's on the mRNA. So this is kind of a summary of the steps from last day. You start off with DNA, which is a double strand of bases that encode for proteins in your nucleus. When you want to express a new protein, then a message is copied onto mRNA, so it copies a portion of the DNA. So the DNA contains many proteins or encodes for many proteins. You'll get transcription of a message for, say, a single protein onto the mRNA. The mRNA will then leave the nucleus, go to the ribosome. At the ribosome, it will translate the message for construction of that new protein or synthesis of that new protein. So to make the new protein, you have to deliver in amino acids, which are the building blocks of protein, and you bring them in one at a time. They're carried by this tRNA molecule, and then you get synthesis of a new protein. And that process down here is called translation. Fill in the blanks review here for the glucose alanine cycle. And again, I have this on a page printed off here, so I'll go over this. And the way I set this up is I kind of set it up a little bit backwards from what I covered last day. So it's a cycle, so it doesn't really matter where you start. But I'm going to start from the bottom here since I've got this listed kind of backwards from where I started last day. So within a muscle, if you're breaking down protein for energy, when an amino acid is broken down in the muscle, its carbon skeleton can enter the Krebs cycle, and that will give you some energy. Now, when the carbon skeleton enters the Krebs cycle, then you're left with this amine group, which is toxic for the muscle. So alanine, which is an amino acid, is formed in the muscle from that amine group that came off of the original amino acid, and it's also formed from pyruvate that you break down in glycolysis. So alanine is formed from NH3 and pyruvate in the muscle. So the pyruvate came from the breakdown of glucose in glycolysis. The NH3 came from the breakdown of an amino acid whose carbon skeleton entered the Krebs cycle. Okay, then alanine would go to the liver and it gets converted to glucose in the liver eventually. But first it's converted to pyruvate. So the amine group gets chopped off, then you have pyruvate formed, and then it gets converted to glucose. And then the NH3 will be excreted from the body, so it leaves the liver and goes to the kidneys, gets excreted, and the glucose will also leave the liver if you're exercising, and it goes back to the muscle to be used for energy if you continue exercising. So that's the glucose alanine cycle. I've got to fill in the blanks here for protein synthesis. So again, I've got the possible answers at the bottom. And again, I've got this here. So the process whereby code from DNA is copied onto mRNA, that would be your transcription. Proteins are assembled from individual amino acids, and this would be on a ribosome. The process by which a protein is built according to the code on mRNA would be your translation. This is a double-sided strand of bases that encodes for proteins and is located in the nucleus. This would be your DNA. And then this leaves the nucleus after copying code for protein from DNA. This would be your messenger RNA, which I have right there. So it will be your mRNA. And then at the ribosome, where you're assembling the protein from individual amino acids, the individual amino acids are delivered to the ribosome by this transfer RNA. So it's your tRNA would be the correct answer to fill in that space. So those are all your correct answers there. Okay, a little bit of a review question here. So what specialized type of cells are greater in people who are responders to resistance training? So who can achieve greater increases in muscle size or muscle hypertrophy? These are your satellite cells. So recall that your satellite cells reside outside your muscle fibers. And in response to resistance training, they increase in number and they also infiltrate the muscle fibers and become nuclei, or a single satellite cell will become a nucleus or a myonucleus in muscle fibers. And that nucleus is where you get protein synthesis occurring. So if you get an increase in number of satellite cells, with training, and those get incorporated as new nuclei or myonuclei in the muscle, you get greater capacity for protein synthesis. Bottom line is satellite cells are important for protein synthesis. You get these other proteins called myogenic regulatory factors that get increased with resistance training. They're also known as transcription factors, so they help to turn on transcription in the nucleus, and that starts off protein synthesis. They're also involved in satellite cell activation, and they are increased with resistance training. So if you want to assess the early changes in resistance training, you can find an increase in these myogenic regulatory factors, or sometimes they're just called MRFs. And they will increase with resistance training quite soon after resistance training. So this is the first thing you see after a resistance training session. Later on, you get increased transcription and increased mRNA. And then eventually you'll get increased amount of protein, say, after weeks of resistance training. Myostatin is opposite to the myogenic regulatory factors, or the MRFs. So myostatin has the opposite effect. It halts satellite cell activation. So it keeps satellite cell activation in check, and it is decreased with resistance training. So with resistance training, you get greater satellite cell activation. So my question here is, what would be the appearance of someone who does not produce myostatin? So you could have a genetic deficiency where your body doesn't produce myostatin or doesn't produce an adequate amount of myostatin. In these individuals, you see unchecked satellite cell activation. And so the appearance would be someone that's very big or someone that's very muscular. So in this